Cancer cell metastasis is one of the leading causes of cancer-related death and is associated with poor therapeutic effects. Cancer cell metastasis usually leads to dramatic changes in cancer cells and tumor microenvironment, and is resistant to current treatments. Therefore, a better understanding of the characteristics of metastatic tumors can help to find appropriate personalized treatments, reduce overtreatment and improve prognosis. Recently, a research team from New Zealand published the results of a pan-cancer study of large-scale metastatic solid tumors in the world's top academic journal Nature. The team used genome-wide sequencing, WGS, to reveal the genome-wide changes in 22 metastatic solid tumors, demonstrating that WGS can play an important role in cancer metastasis studies. The results of the analysis showed that the characteristic gene mutations from metastatic tumors were significantly different. These characteristic mutations could reflect the type of primary tumor. The incidence of whole genome replication, WGD, events was higher in metastatic tumors. In single metastatic tumors, 96% of the driver gene mutations are clonal, and up to 80% of tumor suppressor genes can be biallelic inactivated by different mutation mechanisms. It is reported that the study was led by Professor Edwin Cuppen of the Hartwig Medical Foundation and Professor Emily Vost of the Personalized Cancer Treatment Center. The researchers performed genome-wide sequencing, WGS, and characterization of 2,520 tumor and non-cancer blood samples from 2,399 cancer patients. The median sequencing depths were 106 and 38, respectively, and more than 70 million individual cell variants were identified, including 59,472,629 single nucleotide variants, 839,126 polynucleotide variants, 9,598,205 DNA fragment insertions and deletions, and 653,452 structural variants. A genetic mutation catalog for each metastatic tumor sample was obtained. This catalog complements the metastatic tumor sequencing study in the primary tumor genome database and reveals several interesting findings. For example, this latest study reveals frequent mutations in the MLK4 gene in metastatic tumors, linking the increase in the number of copies of MLK4 to cancer metastasis. The study found that recurrent carcinogenic mutations in metastatic tumors may trigger cancer cell metastasis, which is not present in primary tumors, which was not previously discovered in related studies. Therefore, the researchers speculate that at least in solid tumors, metastatic specific mutations are not the main cause of cancer spread. In addition to single nucleotide variation, the study also analyzed a number of structural variants, including DNA sequence deletions and chromosomal translocations, using WGS. It is noteworthy that the study revealed a universal genome-wide doubling, WGD, event. In some metastatic cancer types, WGD is found in up to 80% of cases, and only about 30% of primary tumors report this phenomenon. WGD is associated with chromosomal instability and is associated with multidrug resistance in chemotherapy. In addition, WGD may provide buffers for cancer cells to counteract the deleterious effects of genomic instability on adaptation, such as destructive mutations and loss of chromosome fragments. To reconstruct how each cancer's genome evolved along the metastatic pathway, the team compared the results with a large WGS study of primary tumors and analyzed point mutations and small DNA fragment insertions and deletions. The results showed that the genomic consistency between primary and metastatic tumors was higher, which is consistent with previous studies. At the same time, the analysis also found that the 10 most common oncogenes in primary tumors mutated more frequently in metastatic tumors. In most cancer types, larger DNA mutations, such as structural variations and WGD, are more common in metastatic cancers. The above results indicate that a hallmark of cancer metastasis progression is persistent genomic instability. In addition, the study used a fine needle biopsy as the primary method of sample collection, which collected cells only from a small area of the metastatic site. The researchers found that more than 93% of the mutations were present in each cell of a given sample. This result is in sharp contrast to previous studies, which have reported higher levels of variation. This extreme homogeneity may reflect the fact that there may be only a few primary cancer cells colonized in metastatic tumors. In addition to similar driver gene and genomic mutations in the primary tumor, the team also found some specific genetic features in metastatic tumors that may be helpful in analyzing treatment or drug resistance responses in individual patients. The team identified genetic variants in 62% of patients in the cohort that could be used to stratify patients to help them receive treatments that have been approved or are in clinical trials. It demonstrates the importance of a comprehensive tumor genomic map for the precise treatment of cancer. At present, it is difficult to detect structural variation using sequencing techniques such as targeted sequencing that cover only a small part of the genome, but because of its reasonable price, it is used more frequently in clinical research than WGS. To maximize the potential of clinical treatment, the research team opened up the data set for the study. In addition, future clinical studies of metastatic cancer may use liquid biopsy as an alternative method of sample collection, while recording metastatic evolution of DNA, RNA and protein levels to further understand the biological characteristics of metastatic tumors and support clinical treatment.